All right, guys, welcome to episode 2 of the Misfits podcast, where we're, talking about the new semester and, our general impressions of week 19 and what lies ahead for the future. So, what do you guys think about Cat 2? We just got the results back, right?
I'll leave you guys to talk about it. It makes me sad. I guess reflecting on it. So I'll
Wait,
leave it
what?
to you
What was
guys.
it making you sad?
Because my results were not what I expected, and so, but based on it, it was a good reflecting moment. It made me reflect on what I did poorly in the past and what I should do better in the future, and how I should, I guess, reformat my general structure and how I approach the content that's being delivered.
Could you
Like, how does
explain
this
that a bit more?
reformatting, how are you going to reformat? Is there like a part of the syllabus that, like, you found that you weren't focusing on enough?
I guess it's more on the idea of like every cat has a substantial increase in the amount of content it examines. So cramming isn't the most wise option in later cats, right? So I guess later cats put more emphasis on trying to be up to date and at least revise content as you're being taught it. So, like for example, even if like in the cat 1, you could probably survive if you just were up to date with the content but didn't bother revising it. You just made notes on it, and then you spent the last last week or last 2 weeks before the cat to properly revise. In cat 4, that's not as effective, because if you're spending the last 2 weeks, is just clearly not enough to be able to cover all the content that's being delivered in the year. So rather, it'd be a lot better if, for example, every week you integrated the notes and the content that you found was important to remember, and then every Friday, or maybe every 3 days, you spent the time to revise it as you were gaining it, right?
Yeah.
How about you guys? What did you guys think?
I'd like to build on the point you raised about not just learning new content, but also consolidating the previous content, because I think we should look past just the cats, right? We are learning material that we would take on for the rest of our lives, being medical practitioners, and we have the responsibility to look after other people. And it's important for us to continuously refresh ourselves on ideas, concepts, and what things are, like how to diagnose this specific disease or how to treat it. We need to be able to look for the signs and symptoms of our patients, and that means staying on top of what we know and also being up to date with new research that will continuously come out, and I would believe is growing by the day. My personal experience with the cat is, I think that I totally agree that we need to be keeping up to date week by week. For myself, I think I did better for Cat 2 than Cat 1 in terms of preparation. However, I think that even still, by not staying on top of the ball for the first couple of weeks of the cat, I was playing catch up for the rest of the 8 weeks, which means I'm having suboptimal results. I think going forward, I really need to be on top of the current content, especially since there's now 2 cats worth of content that we've gone through, which means staying up to date with all these pre-readings and making even more detailed notes, because I've had a look around some of the others, like some of my peers and you guys as well, and I noticed the amount of detail of notes that you guys are writing compared to what I write. It's like, I think it's maybe I'm not putting as much value in making notes compared to other people, because I think that, oh, this point is pretty straightforward, but I think the act of typing it or writing it down helps with consolidating even the most simplest of concepts. Like, for example, what would be the order of food that goes through from the mouth to the rectum and the anus? So I believe it would be like the mouth, then the esophagus, then it goes to the stomach, and then I think the duodenum. Is that correct?
Yeah.
Okay.
Yep.
What comes up?
In the small intestine?
Oh, is it just the small bowel or small intestine and the large?
Yeah, so, duodenum, then like the jejunum, then the ileum, then you go to the ileocecal valve. Oh wait, you gotta talk about the sphincters as well. Yeah, it gets more complicated the more that you think about it.
Yeah, there's like the
Like
internal
as you progress,
external.
there's so many details. Like you might be able to say all the parts, but then it's like, oh, what are the individual features of like each part, and like what are the sphincters that you have to pass through, and things like that. I think it's like very easy to get lost in the detail and then not understand like the broad overview as well.
Yeah, like listening to the lecture earlier this week, I still am wondering, like, how do because food doesn't just go through there, it gets absorbed in like the kidneys and the pancreas, right? I'm not
Wait, what?
like,
Does it get absorbed in the kidneys?
does food get like some
Hey,
of the nutrients, doesn't it go through like the bloodstream or something? Like,
Yeah, as in the
I don't-
small intestine has like a large surface area, so it's able to absorb the nutrients, and that's where it passes from the food into
oh,
bloodstream.
Okay. Well, I guess that's something to look forward to learning about, because I have no idea.
Oh,
How
okay,
do
that's
these
cool.
other organs play a part when food gets digested and absorbed and actually taken up with the organs? I didn't know that. Probably should, but now's the opportunity, right? It's GIT.
Yeah, but actually, something that you were talking about earlier, like why are we even learning this for, right? I think it's something that's really important to consider. Like I remember in the learning and development week, I was like was I in this feedback session? No, I was in the learning professional development record workshop session, right? And then I was talking with these two people next to me. I think their names were like Jessica and Megan. Yeah, but they were talking about this thing called like the professionalist mindset, right? And it's like being so focused on like the results, like that, and like doing like well in terms of academia that you lose sight of like what is like actually important, right? Because we want to like go into the consulting room with a patient and be like confident that we can like care for them, and then they can like place their trust in us, right? It's not about like really getting a good mark on the cat or like knowing all the anatomy like to the minutest detail, right? It's just knowing enough to have like a working understanding so we can put stuff into practice. Because I feel like in reality, like what the most important thing that we're learning right now is probably H&E, right? Because that's the stuff we're actually going to like use in the future to talk to patients and to examine patients. It's like all of this stuff, like the theory, it's important too, but it's all supporting that like practical aspect, right? It's like, yeah, the reason why anatomy is important is because if you have a good understanding of anatomy, then it turns up in your examination, right? Like, oh yeah, I understand that the heart points to like the left, and then the valves are on this side, and that's why I need to examine them like this order. Or the carotids connect to the heart, so that's where you can hear like radiating aortic murmurs like through the carotids, right? Like that sort of thing.
Yeah.
So yeah, I think it's really important to know why we're like learning these things.
I'm a bit surprised you place so much value in H&E. Like, I do agree everything's important, but I place, like, my personal opinion is I place more value in the pathology, because it's like, my one idea I have of being a doctor is like, what is wrong with the patient, right? I think we all need to figure out what happens when the patient is normal, so like the physiology aspect and then the pathology aspect as well, what happens when it goes wrong. So I guess it's more pathophysiology rather than just pathology itself that I think is important. I think that if we're able to figure out what is wrong with the patient, everything else leads out from it. But to get to that stage, you do need to talk to the patient and explore the patient's concerns, and maybe the patient will just tell you what the disease is, right? I believe there was a quote that was mentioned in one of the lectures where if you listen for long enough, or if you talk to the patient long enough, at some point they'll tell you the diagnosis, which I found was pretty interesting.
William Osler. Yeah.
Was it William Osler?
Yeah, but wait, did you guys go to the yarning circle when? Oh wait, remember like the podcast about William Osler?
Sorry, which podcast?
Oh, as in, like we had to do a podcast as like pre-reading for the yarning circle, and it talked about how like Osler, even though he's one of like the founding fathers of medicine or something like that, he actually had like a few like attitudes which were maybe like typical for his time, but also like really like racist basically. So one of them was yeah. Wait, you know what I'm talking about, right, Manus?
Yeah.
I know a bit, yeah.
I know the
like, podcast.
I do know.
Oh, okay. Yeah, but like apparently William Osler, he kept like I think
What year was that?
Huh?
What year was that? What year was William Osler alive?
I'm not sure. All I remember is that he kept the remains of like a Native American woman, I think. And when he was asked to give them back, it was a practice of the time to like keep remains for like study, right? He basically refused, and he basically had like prevailing attitudes during that time, which were not very culturally competent.
Yeah, it was like 19th century, like those type of the beliefs at the time were kind of sketchy to begin with, like the line between what's actually medicine and what's just plain bigotry.
Yeah, that's right. Yeah, it's like race-based science. It's kind of crazy to think that it was only like 100 years ago or so. Yeah. Apparently, the hospital that he established was also like segregated as well, even though segregation had already ended in America by that time. Because I think it was like the John Hopkins Hospital that he established.
That's a big one.
With like John Hopkins, like they were combining American and British medicine, but it seems like they also combine the racism of both both countries together as well. But yeah, it's like interesting how even like our foundations, the Western medical practice, and the heroes that we have, they're like human as well, right? As in, they make mistakes, some extremely large and some which are understandable for the time, but looking back, we're like, how could people have done that? Like these people, in the name of medicine, they did these things.
I mean, even UQ has made some pretty big errors and mistakes in the past as well, in terms of like the cadavers and stuff as well.
Oh, really?
And how I think so. I think that's what they talked about during our intro modules and stuff, right? And then they tried to like return them back, and I guess that sort of explains why they put such a great emphasis on the modules to teach us about anti-racism and ethics and professionalism, so to sort of make up and prevent history from repeating itself.
Yeah, I do agree that looking at our past and learning from our mistakes is pretty important, but we should also be looking ahead. And speaking of looking ahead, our IGP2, the group project, is about these future technologies, right? And the innovations that are currently happening in the medical field. So what topic are you guys doing?
Well,
why don't you start us off? Yeah.
Oh yeah, true.
Yeah.
Sure. So for me, I am working on CRISPR and gene editing, which I think is pretty cool. I've looked at it in the past. Have you guys heard about CRISPR?
Yeah.
Yeah.
Okay, well, just to refresh you guys once more on what CRISPR is about, it's like this gene editing technology that people have found quite recently actually have started experimenting and applying it to clinical trials. And I think it's pretty cool because it's part of the bacteria's adaptive immune system.
Yeah.
Before I did any research, I just knew that CRISPR was something to do with modifying the human genome and basically cuts up some DNA. But looking into it, I found it's pretty interesting that sometimes the bacteria just dies and doesn't get a chance to adapt to the invader that it faces. And the invader in this case would be a virus. So a virus would put its viral DNA into a bacteria, and if the bacteria survives, it uses some of that DNA and it ends up making a CRISPR-Cas9 complex. So what it does is it is able to use the viral DNA injected to target any future viruses with that same DNA. And it cuts up the DNA and basically destroys the virus, which I found was pretty interesting. And then these days, humans have been able to do all sorts of weird and wonderful things, which I find is pretty cool. Like, there are trials of CRISPR being used to treat cancers, which I found was pretty interesting. Like, they're testing whether they can cut out or remove or just edit cancer-causing genes, which I think is pretty cool. Like, what do you guys think?
Yeah, that is pretty cool. Like, I think it's interesting, like, that the mutation for CRISPR came from a virus itself. And it's, like, because the viral DNA basically forms a protein complex that can cut out other pieces of DNA, right? So,
actually, just to clarify, so the bacteria takes the viral DNA and puts it in its own genome, I believe, and then it modifies that to form something called CRISPR RNA, and then that joins to a Cas9 enzyme, which is what cleaves it, just to clarify.
oh, so then the protein, the genes that code for this Cas9 complex are already in the bacteria before the viral DNA gets inserted.
Yeah. There's a Cas9 enzyme floating around.
Yeah. So
I mean,
it's
the
like
last
an immune
I heard
system.
of CRISPR-Cas9 was during undergrad, but I think it might have been during some kind of research. But I think there was like a story a few years back where there was a scientist who used CRISPR-Cas9 on twins.
Yes, yeah.
I think
Oh,
he
yeah.
edited the embryos, and it was an attempt to like make them more resistant or like immune to like HIV or something.
Yeah.
And that was the last thing I learned from it. And then I remember there being like a lot of like global backlash in like the scientific community about like what sort of like a lot of ethical dilemmas surrounding it. Like, what means did he go through? Like, clearly, I don't think this is something that a committee would, an ethical committee or ethical board would have approved, considering how not well understood genetic editing is. And then other sort of cliche topics like designer babies, selection, and things like that. So I was just wondering what your guys' thoughts about like the ethics surrounding like applying CRISPR-Cas9. When do you think? Like, what decision making would you have to go through to be able to apply this type of stuff on people?
Yeah. I feel like CRISPR, because since these genes are probably like, since these genes are being like inserted into these babies, they're probably going to be passed down to when they grow up, right? And then they like have children, then those genes will be passed down as well, right? So CRISPR, by editing someone's genome, you're not just changing the genome of that particular person. you're actually changing the genome of everyone that they will sire like in their lifetime, and those people as well, right? So it can have like massive consequences if like you're too like hasty or you don't understand what's going to happen.
Yeah.
So however, I think you can use CRISPR for like somatic gene mutation, like as in for cells that like won't pass down their genome and like will only replicate in the body, and it's not like a because as long as you don't edit the gametes, then it should be okay and it won't like get passed down.
I think it's sort of oh yeah, Raymond, continue.
Be considered. Like one of my group members has been looking at the ethics, like specifically other ethics. Like I've been having read about it as well, but we came across the two babies that you talked about, Manas. There have been new news, I guess. Like what's it called? Like it's been a couple years since that news came out about the twins, and there's actually been some follow-up on it, and I believe that there have been other new problems actually that have come about because of the genetic modifications. So I think that with genetically modifying humans, it opens a can of worms, and I think there needs to be a lot more investigation before we pull the trigger and say, yeah, let's do it. There's a lot of ethical issues involved. Like, have you guys heard of the movie Gattaca?
Yeah.
They actually mentioned it in one of the lectures pretty recently, which I found was pretty amusing. Because, actually, I'll explain the premise of Gattaca. So, if you have a look at the title, Gattaca, each of the letters are the 4 nucleotide bases in DNA. And that's what the movie is about. It's about genetically modifying people and the consequences it has on society. And what the movie depicts of this concept is there are people who have been genetically modified to have superior traits. So maybe an increased lung capacity or physical appearances or maybe strength, for example. And these people would be considered superior to people who haven't been genetically modified at all. And there's a lot of questions to be raised about what should be allowed to be genetically modified, if we were to even allow it in the first place, because things such as cosmetics, maybe like eye color or hair color, etc., that's one level. But then there's also a question of whether we should be allowed to genetically modify people who we know will have medical conditions as they grow up and when they grow up. I think it is a very grey area, and I'm not fully sure what the right answer would be. Like, what do you guys think about that?
I mean, it's kind of... coming back to what you said about opening a can of worms, it sort of boggled my mind that there's just so much that goes on when the body sort of deals with DNA and genetics as a whole. There's epigenetics, there's polymorphisms. Like, even a slight base change could have like significant ramifications in terms of, most likely it's a nonsense mutation, or if it like completely changes or disrupts the function. So like, thinking about how you'd go about approaching and investigating what the long-term side effects of even the tiniest interactions changing DNA has is like almost overwhelming to even think about. But then again, we're only medical students. I imagine there are more succinct or concise approaches that maybe like PhD or research scientists have taken into consideration. And maybe even that's like where like AI comes into play, or like various, I don't know, I guess data analysis systems to figure out what would be the safest means of implementing DNA. I think definitely safety comes first in terms of figuring out where CRISPR or Cas9 should be implemented, how it should be implemented, who should have access to it, if access should be allowed at all. So yeah, it's definitely just something that's super overwhelming.
Oh yeah, I mean, I think like you guys are like worried a lot about adding DNA to people, like stuff that enhances people's abilities, maybe like intelligence or like strength or that sort of thing. But I mean, what do you guys think of taking away diseases that, like mutations that would negatively affect like a child's lifespan in the future? Like you know that this child will get like, Huntington's in the future. So would you be fine with like genetically editing the child's DNA to just not have that messed up Huntington gene anymore?
I think.
I don't even know how you do that.
Pardon?
How would you take it away? Would you not have like substituted or something with like?
No, that's because I think the Huntington gene. The reason why you get like Huntington's is because they have like too many CAG repeats, like a triple CAG repeat at least in the gene. Yeah. So if you're able to remove some of those repeats, then you can restore normal function to the gene, right?
Oh.
So that's how CRISPR would work, right? You just need to cut it out. I mean, it's not even like you don't need to make a new DNA strand either. You just need to cut out those extra repeats.
Oh.
Okay. I think that what you're talking about does make sense with the CAG repeats. How CRISPR works, to my current knowledge, is that when it takes up the viral DNA sequence or a sequence that humans would want to put in, so I think it's called guide RNA. That's what the human modified version is. If the CRISPR-Cas9 complex were to be made up of a CAG repeat, I'm not sure how specific the Cas enzyme would be, because what happens if it takes out other areas by accident? What happens if it's like non-discriminatory? I believe that's what the right word would be.
Yeah. I think that's why you would kind of like, because I'm pretty sure this has already been solved in CRISPR, but like specifically targeting specific like bits of DNA, right?
Well, it sounds like another area for me to do a bit more research with my group for the IGP.
Yeah.
Do you guys want to share a bit about what's happening on your end?
Yeah, sure. I mean, I can talk about the microbiome bit, which is what my group is focusing on. And I was pretty surprised that this is one of the topics that didn't get picked until the very end. And I don't know. I know that because I picked a bit late. So there were only 3 topics left, and one of them was the microbiome. But microbiome is really fascinating because it actually informs so much of like what our bodies do and like how our body like processes food or like adapts to like certain things or like prevents or our body protects itself against pathogens, right? It's like your microbiome protects you against stuff like C. difficile. Apparently it has links to stuff like obesity as well, which is kind of crazy, and like mood, because the gut has its own like nervous system. Like we're learning about that this semester actually, probably, like the enteric plexus, right? I guess not the enteric plexus, the enteric nervous system.
Oh yeah.
And that also feeds into the brain. So stuff that the bacteria in our gut secretes can be like picked up and affect the enteric nervous system and then affect our brains as well. So it can cause like changes in like eating behaviors. I guess that's maybe how it leads to obesity, and yeah, things like that. Oh, and it's also been linked to autism actually, which is I find quite surprising. Like the actual composition of your microbiome can like decide whether you get like certain I wouldn't say like diseases, but like certain
risk factor?
disorders or not, right? And the thing is the actual like diversity of the microbiome in every person, because everyone has like a slightly different microbiome, because it's the bacteria which makes up your microbiome doesn't just come from like the food that you eat, because then the food that you eat kind of cultures specific bacteria in your gut. So that's why eating fiber is good, because it helps you maintain like a microbiome that has a higher proportion of like healthy bacteria. But it's also the people around you as well, because apparently you can like pass bacteria from your microbiome to other people, like because
how so?
it's because technically when we think of the gut microbiome, we always think about like the stomach and like the colon and all that, right? But the mouth is also an oral microbiome of its own, right? So just like exchanging fluids, like sneezing on someone, I guess, that sort of thing, it can just pass your microbiome to other people, and that can colonize you as well. I think they did a study where they looked at the microbiome of like a bunch of people in a football team, and they all had similar compositions because they just spent so much time with each other, which is a bit strange. And it's not like they're like kissing each other.
But like would
just
that,
like-
would
they're
that
just, like,
spread
angry,
the
right?
microbiome? That is the question.
Yeah,
I think
wait,
that's
what?
the question
What?
on everyone's mind. Like, does kissing spread the microbiome?
I don't think that's the
Well,
question
yeah,
on
obviously it
everyone's
would.
mind.
I imagine it
But-
would, yeah.
Yeah,
Like
I
kissing,
think it does.
coffee,
Like
tea?
sharing food as well.
Okay.
Oh yeah, and also another interesting thing is, like, sometimes people's microbiomes are so different that, like, fecal microbiota transplants, like when you take
Yeah.
someone else's feces, like healthy stool, and then you like implant it into someone else.
Mm.
There was this study where they were like testing FMT's efficacy on like preventing, increasing the remission of irritable bowel syndrome, bowel disease, so IBD.
Okay.
And they found that out of all of their donors, only specifically the people who received feces from one donor had like increased remission, because that donor had like a specific microbiome that was beneficial for treating IBD.
Damn.
So yeah, and then it just didn't work with any of the other people as well. It's just like this one person had like really great people, right?
Actually,
Yeah.
Brendan, could you explain to me how the fecal transplant works? Because I've heard of it, but I don't think I've studied it too much. It's not too well known for me.
Oh, okay. Yeah, I'll be happy to do that. I mean, however, that section is actually handled by another group member, but I'll do the best I can. So with the fecal microbiota transplant, I believe like there's like multiple ways of doing it, but the one of the ways that they talk about in the paper that they give us is they use like a capsule. So then they put the fecal matter inside like a capsule, and then you can either go via the upper GI tract or the lower GI tract. So
What would you prefer, Brendan?
I would prefer the lower GI tract. I don't know.
Wait.
Yeah,
but
I
how
agree.
what do you mean by the upper GI tract? Is that like how is it? Is it like an oral pill or something you take, or is it like some sort of endoscopy?
It's like a
or,
tube.
where they? It's
I
a
think
tube.
they use
so
like
it's-
some sort of like nasogastric tube to like deliver
Oh,
the
right.
fecal matter into your gut, but it just has to get in, like whether it's
So and
one way.
I would assume you'd be under anesthesia when all this
Yeah,
happens.
yeah.
So-
Good,
Yeah.
I
good.
think that's how it
good.
works.
So what would
Um,
you rather, Brendan? Sorry, I mean Manus. You already explained. your
I
point.
would rather take whichever one is best for my outcome in terms of health.
Oh, okay, that's a good answer.
That's a very political answer, I think. What would be your answer if both of them were equally okay? I think I know.
I'd
what
leave it to the
want.
doctor. I'd leave it to the professional medical opinion of someone who knows more about what's going on in terms of my health and whatever I'm suffering from and what options are available and risks and benefits.
Okay, then what happens if you were the doctor doing the transplant? Because at some point we
I-
might be those people.
Yeah,
in
but
that
for
position,
a
right?
patient, for another
What if it was
patient,
for another patient.
well, it would depend on the case, right? I don't know what the patient's details are. I don't know I would have to talk to the patient first and figure out what their preferences might be as well. If both options are equal in terms of benefits, risks, success rate, etc., then I would have to talk with the medical team, as well as talk to the patient, then figure it out together.
Sounds like a good team player. Good job.
As any medical practitioner should, yeah.
Yeah.
Okay, what were you thinking? Clearly you seem to have
I
some
would
sort
have
of
agreed with
idea.
Brendan if I was a patient. I prefer through the lower GI tract rather than the upper GI tract.
Okay.
It's an interesting question to think about. Moving on. Manas, what's your group studying this time?
So, funnily enough, we had our first meeting yesterday.
Okay.
Oh.
And our topic is nanotechnology and nanomedicine. And I have yet to look at any papers associated with it, so I can't say anything about it, since I don't know anything about it yet.
It's early days.
Looks like we're all at different paths. I mean, at different stages in our journey.
Okay. Well, I'll be keen to hear about your project next
meeting, exactly. I'll have something written down.
And
I'm responsible for writing the reflection.
sounds good.
So I'll have a lot to say.
Yeah.
Okay. Yeah, I'm looking forward to it.
Speaking of next week, I think it is about time for us to wrap up this meeting. Well, I'd like to thank you guys for being here with me, reflecting, and I'll see you next week.
Yeah.
Okay, yeah.
Thank you.
Thanks, guys.